Good afternoon, everyone. We're so happy that you can join us today for our webinar with everything going on out there. These are really difficult times for everyone, we're sure, but we felt that it was important to continue with our technology-focused conversation here today. Well, first of all, virus or no virus, spring is here. And with that, pollen season for many of you. And we also know that just like New Tech Group, many of you are involved in supporting the supply chain and are considered essential businesses at this time. And so we need to make sure that we continue to do the best in the most efficient ways. So with this, I'm welcoming you to our webinar about innovative technology solutions for the agricultural industry. My name is Orna, and I will be your host today. Most of you know about New Tech, but I'll share with you just a few words for those of you who do not. New Tech Group is a market leader in implementation of innovative technologies in the quality control and research and development laboratory, in the agricultural space, as well as in food and pharma and other industries. Our goal is to help you, our customers, improve your laboratory workflow, your productivity, efficiency, and consequently, your results. We do this by providing you access to a broad selection of quality instrument solutions manufactured by our strategic partners. Related to our presentation today, we offer cutting-edge instrumentation solutions for pollen and seed analysis, which are used both in R&D as well as in production. Today, you're going to hear from our senior application scientist, Dr. Brady Carter. Dr. Brady Carter is a senior application scientist with New Tech Group. He specializes in quality solutions for the agriculture and food industry. He earned his PhD and MS degree in food engineering and crop science from Washington State University and has a BA degree in botany from Weber State University. He has over 20 years of experience in research and development in both crop production as well as food processing. As an assistant research scientist at Washington State University, he worked with area growers and grain handlers to improve the quality of grain grown in the area. He has authored over 20 white papers, has participated in hundreds of extension presentations, and has given talks at numerous scientific conferences. If you'd like a copy of the presentation after we're done, please reach out to your instrument specialist and they will get that to you. This is a fairly intimate webinar based on the subject matter, and we plan on having some time for question and answers towards the end of the presentation. Don't hesitate to post your questions throughout the presentation and the sidebar. We will do our best to answer, and what we can't answer right now, we'll follow up with you. One last thing before I pass the presentation over to Brady. At the end of the presentation, we'll post a link to a short survey in the chat bar. Please take just a short moment to complete it. We really want your feedback so that we can speak about the topics that you care about. On this note, I will transfer the presentation to Dr. Carter.
Thank you, and welcome everybody to our virtual seminar on innovative technology solutions for the agricultural industry. We're happy to have you here today, and I hope this will be beneficial to everyone in attendance. As Orna mentioned, it's a smaller group for our webinar today, which is nice, and we hopefully will be able to answer any questions you may have, and hopefully we'll be able to cover topics that are of interest to you. So just as an introduction, some of the challenges that folks face typically in the agricultural industry, where obviously the emphasis and the focus is on having effective crop production, whatever that crop may be, and utilizing the land to be able to produce those crops. And of course, when you're in agricultural production, one of the main determining factors for your effectiveness or your success is yield. And yield is, of course, how much product you actually produce, typically per acre of land that's used. And so, of course, low yields are directly proportional to low profits, low revenue. And so some of the challenges that can be faced in terms of yields is when you have these low yields come in part due to inconsistent pollination. And that could be due to several different factors. One of those could be the loss of pollination vectors, as we're seeing this happening throughout the world, where bee populations are in duress, and that their availability may not be what it used to be. It results in sometimes having to have bees brought in, and this can be true of other vectors other than just bees that are sources of pollination. And as a result, at times there can even be this need to preserve now and outsource pollen rather than just relying on nature to achieve the pollination that's needed for good yield. It's become a point where it may be necessary to take intervention steps to try and increase that pollination process. Another thing that impacts yields and production is, of course, disease, or the onset of disease, or coming from pests as well as microorganisms that can attack the crop itself. In order to be able to effectively intervene with the onset of disease, you need to know as early as possible that it's present, and try to have your interventions the most effective at times when the crop can still be saved. In addition, crops now are increasingly have more stringent quality specifications and requirements, especially because most of these crops that are produced become ingredients for other food products. Seeing an increase in demand in terms of consistency for those ingredients, as well as the safety of those ingredients. In one way, especially when you're dealing with commodity types of products, which is a lot of what these agricultural products are, you tend to get the price that's associated with the commodity. One way to be able to try and increase that potential return, or the potential price you can get, is to have value-added traits. And that's a very popular thing now, is to try and create a unique type of product, and as such be able to get a higher price. But in order to do that, you have to be able to capture that value-added trait. You have to be able to show that it exists, and in a lot of cases certify it. And then, of course, for future production to respond to changes in the environment, changes in demands, there has to be effective development of new varieties of crops. And so it's critical to be able to facilitate that process in order to overcome that challenge. So today, real quick, we want to talk about some of these innovations that in technology that will assist in being able to overcome these challenges. And what I'm showing here is just a quick overview of what New Tech does, the sponsor of this webinar. It provides solutions for many different applications throughout the food production process, as well as pharmaceuticals. And you can see just a quick overview of what some of those solutions are. But in particular today, we want to focus in on two of those solutions that have particular application in agricultural production. That includes the Videometer, Multispectral Imaging System, and the Flow Cytometry pollen analysis system that comes from Amphasys. So first, let's talk a little bit about pollination and its importance, and maybe some solutions that we potentially can provide. Of course, all fruit-bearing crops require pollination in order to produce the fruit. This pollination process and how it's done, of course, is variable depending on the crop. Some are self-fertilizing, some require the use of vectors. Some use wind as the vector. And so there's a wide variety of possible ways that pollination occurs. But in all cases, pollination is directly proportional to yield, which of course is directly proportional to your revenue. So successful pollination is critical in terms of fruit production. And so the key elements of that are, of course, that you have viable pollen. If the pollen that's applied is not viable, then obviously that's going to have a reduction in your potential yield. And another factor is pollination vector availability. Are there things that are impacting that? And are there ways that you can overcome maybe those deficiencies that might exist to try and keep or maintain a high level of pollination? And pollen viability, of course, is impacted by a lot of different factors, including the time of day, temperature and humidity, the presence of pesticides, and each of the different varieties of crops, their pollen has its own unique characteristics, including its size and shape, but also in the factors that it's most susceptible to. So there requires a lot of specification in terms of how to improve this pollination process. But ultimately, what you're trying to find out is how to distribute viable pollen in the most effective way. So some applications for pollen optimization. Of course, one of the main ones is to optimize the amount and quality of pollen that's being distributed and coming into contact with the flowers themselves to lead to the process of fruit development. And so that may require, and in some cases is made more efficient by collecting pollen manually and then redistributing that pollen. That comes with some challenges, obviously, but key and critical to that is to know and make sure that you're maintaining pollen viability throughout that process. Now, in terms of collecting that pollen, there are optimal times of collecting the pollen in terms of time of day or particular timing of the year. And key to being able to optimize that process to make sure that you're getting good viable pollen is to track its viability. And then, of course, if you're going to apply pollen manually, you want to make sure that you're applying pollen that's actually viable and has been tested and make sure that it's going to work. If you're going to store pollen for any amount of time and then transfer it, what are the best methods for doing that? And that takes some experimentation to figure out what the optimal processes are. There is a fair number of examples of in the literature of optimal ways of treating pollen, depending on what the pollen type is. Again, it's very specific to the species, but there is some information there. But of course, to optimize your storage and shipping, again, comes back to tracking the viability of the pollen. If you're going to do any kind of difficult crosses where pollen viability is going to be critical, that you don't want that to be the source of failure when you're trying to make these types of crosses, since there's a lot of different factors that are going to be problematic. One that you don't want to be a problem is that you're trying to use pollen that's non-viable. As well as the result of those crosses, the next generation, is that pollen that's produced from those crosses going to continue to be viable? And that is critical to know as well. And then as we move towards higher temperatures, a tendency towards higher temperatures, being able to observe and study the heat tolerance of pollen will be critical. All again, coming back to the viability of the pollen. And then, of course, as well, how are you going to protect it? Is there ways to protect this? And how might you be able to do that effectively? So here's just an example of what a pollen testing implementation scheme might look like. So here are the ideas that you're going to try and collect pollen and then redistribute it in order to maximize efficiency of your growing process. So there are obviously a lot of different steps during this process of collecting pollen. So you have your growth, and then you're going to collect it. So here's a place where you want to try and take a quality test and try to figure out through experimentation, when is the ideal time of day? When is the right flower maturity, and how do you collect this pollen? All of those are going to be factors to be considered, and the ultimate way to check whether or not those things are effective is through checking the pollen and its viability. So these quality gates become everywhere where they're inserted is basically a place where during this process you're going to want to check pollen viability to make sure that things are being done in an optimal way. Now, once they've been established, they can become standard operating procedures. But even when you've established a standard way, you're going to want to check it constantly, and even to the point where if you're going to distribute this pollen as part of a business plan, it'll be critical to have some sort of a certification that that pollen is viable. And so again, as you move along the process to storage, now how are you going to store it? At what humidity? At what water activity of the pollen is it going to be stored at in order to maintain maximum life? How are you going to restrict light during that storage? Are you going to freeze it? If you're going to freeze it, what's the most effective way to freeze it and still maintain viability? And then, of course, you're going to, so you want to make sure that after that storage process, before you send it out, you still have viable pollen. Now you got to transport it. What's the best way to transport it? You want to make sure that your methodologies for transport are effective. And so again, the way to do that, check for the viability. Then you might have to rehydrate it before application. Is the rehydration process effective? And how will you check that? Again, here's another point where you need to check pollen viability. And then you'll have the final pollination step, and then connect that in the end to any types of increases you see in yield as a result of this process. So some examples of pollen viability case studies where this could be utilized. Of course, in a hybrid crop production, you have to have pollination from the male hybrid parent to the female hybrid parent, and typically that's just done sort of by placement in the field to try and achieve that. But through interventions, it's possible to improve the efficiency of that by manually applying that pollen. But that requires to collect it, store it, and then redistribute it. And how is that done? What is the impact going to be on the viability of the pollen? And so that's a case where you would want to be tracking pollen viability. Another might be where you're producing fruit, where pollination is critical, and you might try to increase yields by manual application of the pollen to the fruit trees. And you might do this by simply distributing the pollen to the vectors to have them distribute it. If there's vectors are a problem, there are applications where that pollen can be distributed using artificial vectors, such as drones, where these drones are actually utilized to distribute the pollen where it needs to be distributed. But again, if you're going to use that type of an artificial vector, it's critical that the pollen that comes or is distributed is going to be viable. Otherwise, it would be wasted efforts. And then finally, are you going to store that pollen and possibly freeze it? What are the ideal storage methods? For instance, there's a reference here for a paper where they looked at storing dried corn, and critical was what was the water activity at the time that it went in to storage. Also, information out there on freeze drying and whether freeze drying is effective, and if you do freeze dry, how do you need to freeze dry, and how do you need to bring it back out? So there's a lot of sources of variation in terms of making this optimal, and so it makes it difficult to give estimates of exactly how much the viability will be decreased using these processes. In this particular case, they said they saw a retention of about 50% of the viability, and so that's going to vary, obviously. The key then is to be able to, if you're going to do this type of work, is to have some methodology for being able to track that viability so that you can identify what is the most effective method. And again, it's going to be particular to the pollen type, the species, and the type of steps you take. And so it really comes down to doing that kind of experimental work yourself. But key to that is going to be to have a way of being able to check pollen viability. So I just want to show this is an example of a case study use for a company called Power Pollen and video is used with their permission. But it just as an example of one company who has taken this idea of trying to improve pollination to try and improve yields, and how they've implemented a technology from Amphasys that allows for the ability to check pollen viability. So what you see here is the collection of the pollen. This is corn, of course. The machine is out collecting pollen off the male hybrid parent. It's going to take that pollen in and be able to store it now, and this person is now going to check it and determine whether or not that pollen is viable. So the system you see in the back is the technology from Amphasys that uses flow cytometry. And then once it's been checked, then it's going to be reapplied back out to the female hybrid parents directly onto those to result in higher yields and higher seed quality through the control of pollination, or the pollination process. Key and critical to all of this is to make sure that that pollen that's being delivered is viable. And how is the process of collection, the process of storing, the process of transport impacting that viability? And the way to do that, and the way that Power Pollen does it, is through using this technology, this flow cytometry technology from Amphasys, to track that viability. But these applications go way beyond just in hybrid corn. This would be any scenario where you're trying to improve yields through the control of pollination, or if you're trying to set up a business plan where you're going to help the growers through the delivery of viable pollen. So here is the technology then. We've talked a little bit about how this could possibly or potentially be used. And now we're going to spend a little time talking about the technology itself and how it works. There are various ways of looking at pollen viability. The traditional ways are fairly cumbersome, require staining and different kind of laboratory methods, can be very time consuming, very laborious, and produce results that are somewhat variable. So the system that's been developed by Amphasys is an effort to try and overcome some of those limitations and make this instead into a fairly automated, straightforward, and fast process to be able to check pollen viability. And the concept behind this is that if you take cells and you pass them through an electrical field, and so this system is set up where you basically suspend the pollen in some type of a carrier buffer where that's dependent upon what type of pollen you're working with, and then that fluid is transferred through a chip that contains a flow chamber that sits in between electrodes that create an electrical field. And as a cell passes those electrodes, what will happen is that you'll see a disruption in the electrical field, an impedance in that electrical field. And so every time that there's an interruption is an indication that a cell has passed. And so as a result, the system is able to count every time a cell passes. But even more importantly, it can differentiate the condition of that cell based upon that the impedance of the cell in that electrical field. Cells that are intact and have their cytoplasm in place and their membranes will have a different level of impedance than will be cells that have died and are no longer viable. And so the system is able to both count the cells or the pollen, but it's also able to count and differentiate between pollen that is viable and pollen that is non-viable or dead. So if we watch just a quick video, this is an example of how this process works. So within the chip inside of the system, these pollen cells flow through the use of a peristaltic pump and moves the liquid through, passes it through the electrical field. And as it passes through, there's a disruption or an impedance of the field that's then tracked and measured. And through that measurement, it can track then both counting and as well as what is the particular viability of that pollen cell. So the results that you'll get basically look like the graph that you see in front of you, where you have uh, an image or a graph that's plotting the electrical response of each of the cells that's gone through. And so it's tracking them based upon whatever that electrical response is. And in this scenario, everything that appears to the left of the line is identified as dead, and dead cells will always congregate to the left, while viable cells will always congregate to the right. Now, the exact point where that differentiation occurs is dependent upon the pollen and the scenario you're working with. What's nice about this system, though, is you can very easily, just visually, determine where that distinction point is. If you have two specific populations forming, the one on the left is always the non-viable. So you can visually put in a line, and that's what this vertical line is called a gate, and it is an indication of the differentiation between pollen cells that are dead and those that are viable, and it will even give a report. So in this case, this fresh pollen, there was 80% that were viable and around 20% that were dead. If you see a single population, that can be more difficult to figure out whether you're dealing with dead or viable. And so in those cases, what you can do is actually do an inactivation step on the pollen itself through a heat treatment, and then read that to see whether or not that matches up in your case with whether your cells fall to the right or on top of that population that's created through the inactivation step, to create and form in that case where that vertical gate should be. And so through this process, once you've established where that gate is, it should stay fairly consistent in terms of your particular type of pollen that you're analyzing across multiple samples. So from that point on, it's just simply about a 5-minute sample preparation to suspend the pollen in the fluid. You put it into the instrument. The actual analysis in the instrument is a matter of maybe 20 seconds to be able to complete, depending on what type of reading or what pollen you're reading. But it's a very fast test in terms of the actual test with just a little bit of sample prep before. In addition, this system can be taken into the field. So these tests can be done right in the field. There's a battery that can be fitted to the system and allow you to put it on the back of a truck and take readings right in the orchard if you so choose. So an example of how effective this system could be is let's say you were to take the system and put it on the truck and go out into an orchard, and where you're going to take samples of pollen to try and figure out when is the optimal time for capture of pollen. So if you were to do that in this example, you were to take it at a particular time in the day and where there was a certain temperature and light, and you recorded that. Take that sample, you put it into the machine, and what you see is you get a fairly high level of viability at that point in time. You then take it again at a different time of the day, and now you see mostly just dead pollen cells. That's an indication, and then tells you that it's critical what time of day you're going to collect this pollen, as well as the temperature, the humidity, the light, so that you can then use that information to optimize and make changes to how you're going to collect the pollen going forward. So, of course, the ultimate goal of all of this is to increase yield. So the question is, if you do have, if your pollen that you're applying shows higher levels of viability, does that translate into higher yields? And it does. So you can see an example here of a study that was done where the pollen was checked, and you can see on the bottom that it's all showing up on the non-viable side, and you're seeing almost no yield, versus as you increase the concentration of the viable population, you see a subsequent increase in yield. So this process of ultimately trying to achieve and assure the pollen viability will translate then into higher yields, which will then, of course, translate into higher profits. So here's just a quick partial customer list of folks that are already interacting and working with this type of technology of using flow cytometry to track pollen viability. Okay, with that, I want to shift real quick away from our pollen discussion to more focused on when the finished product, what you actually produce, and how the technology that we provide can be helpful in terms of the actual product and the processing of the product and the handling of the product after it's been produced. So some examples of what I'm calling non-conforming ag products. So these are products that are not meeting the requirements, the specifications, the expectations of what this product should look like when it's produced. So what are and of course when that happens, that reduces the value of that crop. And so what are some things that could potentially happen that could result in a product that would be considered non-conforming? So, and we mentioned this earlier, but a big one is disease infestation. So this can result in there being presence of mycotoxins in a product. It can be from loss in biomass that can lead to loss in yield, or if the biomass itself is the product, then of course it's going to be unacceptable if there's disease. Other examples are plants that have poor health and no intervention is being made, then it will produce low yields. If you don't have the capability within your production process to sort your product and eliminate non-conforming products, then the presence of that, whether it be an adulterant, and whether that adulterant is there because of just processing, or if it's even there as a result of handling by someone else in an effort to what's called revenue-driven adulteration, where you're basically trying to fool somebody by putting something in that's not supposed to be there and still get the same price for it. And so it's important then to be able to sort those products and identify what their the unique characteristics for those. And that leads to then the final one, which is if you are going to put a value-added trait into your product, such as a unique shape, color, nutritional value, or any other particular trait, in order to be able to get a higher price, that must be segregated and identified as being unique. Imaging is a great way to be able to identify and segregate any of these non-conforming products, whatever reason it may be. Imaging is a technology that can be very helpful in being able to achieve these desired results. So for instance, we're talking about disease detection. So if you're dealing with some type of whether it's grain, whether it's biomass, whatever it is that you're producing, if you have the earlier you can detect the onset of disease, the better chance you have of being able to use some type of an effective intervention, whether that be the application of a pesticide or other interventions that you might do. What imaging allows you to do is to be able to detect very early on the presence of any kind of disease, even before it's necessarily possible to see visually. So the example here is on some grain where you can maybe see some indications on the normal image of being able to see that something might be happening. But when we use image analysis and we create basically what's called a heat map, we can use that image analysis to very clearly identify exactly where the disease is present and even be able to figure out what percentage of the product has disease, and ultimately even sort and remove disease product if so desired. So this allows for interventions that can be done more effectively and earlier in the process. In terms of plant health, if you do traditional color imaging on a plant to look for chlorophyll, which is a great indicator, obviously, of plant health, and you have two types of chlorophyll, A and B, and you look at an image of that using just the visible range. Chlorophyll A and B in that range look fairly similar if you were to look at their spectra. So that makes it difficult to figure out if something's going on, as one could be hiding the lack of the other. With spectral imaging, specifically multispectral imaging that we're talking about in terms of this technology, by being able to extend the image analysis into the UV as well as up in the near infrared, makes it possible, using more of these wavelengths, to differentiate between the two types of chlorophyll, so that you can get a better indication of what the plant health is, in particular to the product that you're working with. Now, in terms of seed purity, if you're producing seed that's certified seed, of course you need to be delivering the seed that you say you're going to deliver. But in addition, if the seed itself is the product that's going to be distributed, the presence of adulterants can be very dangerous. For example, if you were to have a piece of peanut in these sesame seeds, and then they get used to make some type of a product as an ingredient, and that peanut goes along, and then someone with a peanut allergy consumes it, then that can lead to a potentially dangerous situation. If you were to look at this image visually, if you took long enough, you might be able to see that something's there that's not supposed to be. But when you, again, look at an image analysis and take a look at the heat map, very clearly from that analysis, you can see where that particular adulterant is, and even would have the capability, again, of being able to remove it. So in terms of assuring seed purity, this image analysis is very effective. It's also effective at being able to determine the purity of seeds. For instance, in this case, we're talking about spinach seeds, very high value types of seeds. So when you're distributing that, especially as someone who's buying that, they want to know that they're buying actual spinach seeds and not weed seeds that are being mixed in. This system has the capability of being able to differentiate between what is a spinach seed and what is a particular or different kinds of wheat seeds based on shape, color, size. So it has that ability to then tell what is the percentage of purity in terms of this seed lot. And if so desired, it can even then be used to remove those weed seeds specifically by sorting and removal. The system can take seeds of any type and basically show for each individual seed its particular characteristics, its shape, its size, its color. It can sort by those colors. It can so for instance, in this case, these are sugar beet seeds, and by doing this sorting, you can see that are appearing in this group are several different fragments that are not going to have any kind of viability. And so the way it would be possible to then purify this seed simply by removing those fragments. But in addition, you can see a distribution of the different shapes, the different color types, and get an idea of whether or not this is going to be a viable lot of seed, or if maybe it needs to be thrown away, or if there needs to be further segregation done on it. So this obviously would allow you then to segregate and differentiate by specific traits, whether that's, again, shape, size, color, maybe another particular trait. The beauty of this system is that it can be taught to identify and recognize any particular trait of interest and then sort by that trait, even not only from a physical standpoint, but even from a chemical standpoint, due to its ability to look in both the UV and the near infrared range. Here's another example where we're dealing with cranberries that have to have a particular firmness. The ones that are too soft go to juicing. The others go to either fresh cranberries or into drying and sugar infusion. So it's a critical attribute of these cranberries. And so the earlier on that you can eliminate those that are not conforming to that desired firmness, the less waste you produce, and the higher value that you can have for the finished product. The system can differentiate each of these different cranberries by their firmness level and sort by them, and then allow for selection or sorting based on a firmness. Here's an example where you can, another example where you have two different wheat types that could potentially be mixed, which would make them basically an adulterant in the other one. You don't want hard wheat and a durum wheat that's being used to produce primarily pasta versus a durum wheat that you would have in a hard wheat that's being used to produce bread that has particular gluten strength properties. So any one of these being mixed would be considered an adulterant. And this system has the capability of being able to very clearly differentiate durum wheat from hard wheat, even though visually it may be very difficult to differentiate between the two, especially without very specialized training. This system can very quickly capture those differences and allow for determining whether or not a contamination event has occurred. In addition, for grain, one particular problematic process that can happen is called pre-harvest sprout. For those who deal with grains, they're probably very familiar with this, where the sprouting or germination process basically begins in the seed due to typically to exposure to high levels of moisture through a rain event. When this happens, it basically renders that grain unusable because of this germination process starts to break starch down. So being able to detect that is critical to making sure the crop is good and not mixing in grain that has experienced pre-harvest sprout. Currently, that's tested through something called the falling number, which is a wet chemistry test that can be fairly labor intensive. Imaging would be able to do the same thing, but without in a non-destructive and very quick way. In addition, if you prime seed, the priming process is meant to essentially start the germination process and then freeze it so that you'll get better germination out of the seed itself. So critical to that is knowing when the germination process is started so that you can then freeze it or stop it through treatment and freeze that process in place so that when that seed is then planted, you'll have an increased level of germination. So I want to show you some examples of how this has been effectively implemented in different scenarios. So this first one just shows an auto feed system teamed with the technology. The technology you see there is the video meter. As it goes under the video meter, it gets a description applied to the robot, and the robot is then capable of being able to sort based upon whatever particular trait of interest is coming from the imaging system. So the imaging system's taught to recognize a specific trait and differentiate by it, passes the information on to the robot, and then the robot sorts based on that information. And so the result is that you have very pure products that come out from that. Here's another example of its utilization in a greenhouse scenario, where the idea here is to track plant health over time with a very complex experiment. Through automation in this greenhouse, they've automated the process for watering and of course delivery of any kind of interventions, fertilizer, whatever the case may be, in an automated way based on a grid system. So each one of these plants goes through that process and receives the watering. And then after this watering step, it moves into another chamber where some imaging can be done, including imaging done with the multispectral imaging system. So in this next step, you'll see the system come down over the top of the plant, takes an image, records that image, and then saves it according to the grid location, which can then be tied directly to that particular plant, and tracked over time will give an indication of any changes in plant health that are happening on that particular plant. Next, we have an example of utilization of the system right in the field. This, of course, is a benchtop type of system, and in its off-the-shelf version, but it can be engineered in, as you've seen, in many different ways. To be used on a production line, to be used in a greenhouse environment, and to be used in the field, where basically have designed this analysis system that allows for to be attached to a tractor. Inside of it is contains the multispectral imaging system. They take it out into the field, drop it over a plot, take an image, continue to do that over time, and that allows you to then do an assessment of plant health, and then which could be then critical in making selections within those plots. So what is the imaging that's used here? Again, this is multispectral imaging, a very specific type of hyperspectral imaging that basically takes an image at different wavelengths covering, again, from the UV range all the way to the near infrared. And so it essentially collects 19 to 20 different images, and on each of those images, a pixel then, a particular pixel is represented on each one of those different images, which allows the system to be able to do statistical analysis on a pixel by pixel basis across multiple wavelengths to try to look for and identify any differences of interest. And so through machine learning, if you identify to the system a unique particular trait and differentiate that from another, that being the control, and then a variation of that trait, the system can then use that to recognize the differences in those particular traits in future unknown samples. And it does this in a non-destructive way. The system just drops down over the sample, takes an image, and then comes back up, and your image is then ready for analysis. Very little sample preparation needed. And the analysis itself takes about 5 to 6 seconds. And again, what you get from this is in a multispectral imaging or image, you're going to get an individual images representing wavelengths all the way from UV up to near infrared. You can even look at each one of those images at different wavelengths individually. The system can then combine them into one image that looks basically like a typical image that you would collect. But then the power comes in the software's capability of being able to take each on a pixel by pixel basis and do analysis on each of these images, the combination of the images, find the correct combination that does the best job of differentiating based on how you teach it, the information that you give it in terms of the distinctions that you want to look at. The system then takes that information and finds an optimal way of being able to differentiate or identify your particular trait of interest. So here's an example of what those images would look like, going from all the way from the again from the UV range down at 375 nanometers all the way up to the near infrared range at 970 nanometers. This is in a grayscale view. You can also do this with a heat map view, back to those images we saw earlier where you saw red and blue. But basically what you can see here is that as you move between wavelengths, different parts are highlighted of the grain, showing physical differences. But again, the system has the capability of locking in on chemical differences. Now, this is done from a surface imaging, so it's not able to look in at particular chemical attributes inside of the seed, but or inside of whatever product you're looking at, but more from a surface detection standpoint. So it's not. It can be taught in certain cases to see differences in things like constituents like protein, if there's a manifestation of that's fairly on the surface, but is different from, say, a near infrared analysis where you're basically trying to do a spectral response and link that to a particular trait of interest. In this case, instead of doing that type of analysis, we're looking at trying to utilize the information across multiple wavelengths to lock in on distinct differences and use those to identify those differences to be able to differentiate and sort in future unknown samples. So in summary, of course, agricultural production has unique challenges associated with it. Those challenges are made more challenging by certain events, whether that be the type of things we're going through right now, changes in environment or in growing conditions year to year. And but of course, the profitability of any agricultural production process is to minimize the inputs, get your best price possible, and maximize yields. And when you can combine those, then you get your maximum profitability. It makes complete sense. But how do you minimize the cost of inputs? How do you get the best price? How do you maximize yields? And hopefully, the technologies that we've looked at today, you've seen examples of how they could be utilized and implemented, whether that's to detect disease early on, to minimize the types of inputs that have to be put in, to track plant health, to know exactly when any inputs would be useful, to make sure that the seed that's being used is optimal, and of course, whether the pollen that's being applied is optimal as an input. And then once the product's produced, we've shown examples of how the technology can be used to segregate um, based on a particular trait, or to identify adulterants that cause the price to come down or for the lot to be rejected, using these types of technologies. Um, but again, the capabilities of these systems, to be able to detect viability, to be able to sort based on particular traits, is extremely wide. And so what we do in most cases is that when we interact with someone who's interested in utilizing this technology, we do take it on a case by case basis where we spend some time. A lot of cases we have people send samples in, whether that be pollen or seeds, for us to be able to do some analyses on as examples, and then share those results and talk about what the results mean and how it could be effectively implemented in a case by case basis for each unique situation. Because again, these tend to be very unique in the way that they're applied, depending on the particular application and what needs to be done. And that is part of my role at New Tech Group, is to help with that process. So I'm here to assist anyone who's interested in how you might be able to apply these technologies. We don't just give you the technology and expect you to figure it out on your own. We're here to help and provide support, and we really believe in these technologies and think that they have a huge amount of potential to help with agricultural production and overcome these challenges. And we hope that that they will be beneficial, and that we might be able to, in some way, be able to help you overcome any challenges that you might have. And with that, I'll leave our contact information. And again, there's included here is the email for our sales group. If you're interested in getting a copy of this presentation, please reach out to that email, or if you know your particular sales rep, they likely got in contact with you prior to this webinar, so you should have their email address to reach out to them. I'm happy to set up conference call to make do some discussions as well, as a follow-up to this webinar, especially if we want to discuss specifically your particular applications. And with that, I'll turn the time back over to Orna.
Hey, Brady, thank you so much. Take a breath. There are a few questions in the chat box. Do you see them, or do you want me to read them to you?
No, I can see them. Let's see. One question that came was whether or not you could sort dead pollen from viable, so that you're only collecting viable. That is fairly challenging because the pollen is so small. Certainly, though, if you were to, if you had a particular environmental effect that could result in, or a timing or whatever, in a high level of non-viable, having the system to check it would give you the capability of knowing, especially if you had it, say, out in the field, and you were checking, and then you started to see viability drop off. It would give you then the capability to stop collection, or to not utilize the pollen that's been collected in that time frame, to try and keep as much as possible the non-viable out, but it would require a higher level of testing in order to be able to identify when that the pollen is becoming non-viable. Otherwise, if you collect it as a whole, once it's been collected, it'd be very difficult on a pollen by pollen basis, obviously, to differentiate that. Would require probably use of a microscope, and that's not necessarily. It's something that we have started to look into through the use of imaging on the imaging side, where that may be possible to sort on a pollen by pollen basis using a specific version of that imaging, which is called the Videometer Micro, where it utilizes imaging along with a microscope. Don't have At this point, we don't have a particular case study showing that actually working, but there could be the potential to do that on even on a pollen basis if you were able to have some way of being able to differentiate the viability and then knowing that it's in a particular location. Again, a lot of challenges associated with that. Would be much more feasible to be able to do it on a larger lot by lot basis, certainly through testing and isolating on the ones that have the highest level of viability. To that end, there's the next question is whether or not the imaging system could be used in tandem with the viability determinations. Again, back to that same question. There is that potential. The resolution on the imaging system as is tends to not be good enough to be able to differentiate pollen that's so small, but with the micro version, it could be possible, but we don't have any specific case studies of that at this point. Okay, we've got another one about what is the particularly the prescribed protocol for pollen storage and how shelf life affects viability. How does it change over time? And I mentioned some examples of studies being done to show that. The problem is that there's no general protocol that's going to be the best and ideal for everybody. Very specific to the particular environment that you're working in, the type of pollen that you're working with. And so being able to establish what are those best practices for a particular type of pollen requires some investigation up front. And again, key to being able to do that investigation is going to be to have the Amphasys system. So I'm happy to help with setting up that type of an investigation. It's just not something that's sort of a magic bullet for everybody that this is going to be the best way to do that. There are some general rules that can be obtained, and I'm happy to share that on a case-by-case basis since it's, again, on a specific to the pollen itself. But certainly figuring that out is possible using this system and to get a good idea of what that is for a particular type of pollen in a particular scenario.
Thank you again, Brady, for this wealth of information. Before we wrap up, please look at the chat box. You can already see the survey link. Let us know if you'd like someone from our team to reach out to you if you're interested in having a copy of the presentation or for any other information. New Tech plans to be open for business and responsive. We consider ourselves an essential business in support of essential industries, and we're here. Thanks again for joining us. Stay healthy and stay safe, everyone.